Hey everyone! I don't know about you guys, but I've been struggling to find an efficient way to review anemia. So I'm making this video while I'm rewatching the actual Panapto lecture so that I can quickly summarize the pertinent information. And I think we're all pretty comfortable with the common signs and symptoms, so I want to point out a couple of these key lab values. The first is mean corpuscular volume. That's going to tell us if it's microcytic, normocytic, or macrocytic. And one of the key things to remember if we look down here is it's actually measuring both the reticulocyte and the erythrocyte average corpuscular volume. So you might have a situation where your erythrocytes are smaller than average, but because the reticulocytes might be a little bit bigger, the overall average of the two put together could land you in the normocytic range, and that'll be a little bit clearer when we get into the different types of anemias. Another key one to point out is the MCHC. That's the mean corpuscular hemoglobin concentration. And I think we're all pretty comfortable with this now. That one indicates hereditary spherocytosis when it's high. Red cell distribution width measures the variation in red blood cell size. So if you have a lot of small and a lot of large red blood cells, then that's where you're going to have a significant red cell distribution width. We're also going to get into HE in a second here. So all of these nucleated cells, this entire process of precursors to red blood cells all happens in the bone marrow. Once you reach the peripheral blood on a normal under normal conditions, you're just going to have the reticulocytes and erythrocytes. So if you were to have a slide that did have some nucleated cells mixed in here, that would indicate to you that something's going wrong. Maybe there's an anemia condition and the bone marrow is trying to compensate, so it's trying to quickly push red blood cell precursors out into the peripheral blood, even though they're not fully developed and have not released their nucleus. Another thing that you might see if you do have anemia is maybe a reduced amount of reticulocytes. And if you have fewer than normal reticulocytes, that's when you would start to consider that it's more a production of red blood cells that's an issue rather than like a hemolytic anemia, for example. The reticulocyte hemoglobin equivalent, RET-HE, is a measure of the relative amount of hemoglobin in your reticulocytes, and it's indirectly a measure of iron. So this is a quick test that you can often find inside of the CBC, and it can tell you that you either have an IDA, iron deficiency anemia, or an AI, anemia of inflammation. It's not a specific test, so this is fast, this is a starting point, but then after this, if this comes out low, you might have to order an iron panel after this, which would take a little bit longer. And then just looking at the slides here, I wanted to point out the reticulocyte is this a little bit more purple one, and it's a little bit more purple because it has RNA in there. You can kind of see that a little bit up here as well. And then with this different stain, you can see the reticulocytes are here with their RNA. And then remember, the 1/3 central pallor is sort of a textbook thing. So in reality, you might not necessarily have exactly 1/3. Some of them are going to be a little bit more pallor, have a little bit more pallor than others. This slide comes out quite a bit, but I did want to point out the difference between the reticulocyte being greater than 2% and less than 2%. So hemolytic anemia is where somewhere the red blood cells are dying faster than they should. And in order to compensate, the bone marrow is trying to push out a lot more red blood cell precursors and reticulocytes than normal. And so that would cause us to see a high reticulocyte count. So hemolytic anemia, high reticulocyte count; non-hemolytic anemia, normal or low reticulocyte count. And remember, these are also the other end of things, right? So if you have iron deficiency, then you're missing one of the key ingredients for the creation of hemoglobin, which is the key ingredient for the creation of red blood cells. So we would expect to see a lower reticulocyte count or sometimes a normal reticulocyte count, but we would not expect to see, if we're missing some of the key ingredients, an increased reticulocyte count. Right, so reticulocyte count can kind of point us in the direction, is it a construction problem or a destruction problem? Moving on to macrocytic anemia, so that's, remember, referencing the mean corpuscular volume, in this case greater than 100. So some of the key things here are going to be vitamin B12 and folate deficiency. Vitamin B12 and folate, think of them as ingredients in the production of red blood cells. So if you don't have the proper ingredients, then you're not going to have production or as good production of red blood cells. The term megaloblastic anemia refers to these megaloblasts, these cells in the bone marrow being larger than average, and then macrocytic is referencing the actual red blood cells, which in this case it doesn't give us any white blood cells that we can compare, like the red blood cell to the nucleus of a lymphocyte. But it's trying to show that these are larger than normal. And then another key finding in this type of anemia is more than 3 to 5 nuclei lobes in a neutrophil. So it's essentially a deficiency in vitamin B12, and there's a lot of steps in the process that can occur, so I'll let you review that on your own. I'm going a little bit quickly through this because she said at one point that we weren't really going to be tested on this, but I wanted to discuss pancytopenia is going to be low red blood cells, low white blood cells, and low platelets. That's the definition of pancytopenia. And then pernicious anemia is another term that comes up sometimes, and some indications are going to be a high gastrin. Folate doesn't have the neurological symptoms that B12 has, so if you have these slide findings and you have neurological symptoms, it might point you more towards vitamin B12 deficiency, so you could test that. Next, we're going to discuss some of the normocytic anemias, beginning with anemia of inflammation. If we see used to be called anemia of chronic disease, and chronic disease is a major source of inflammation, but it's not the only cause of inflammation. I want to highlight here this hepcidin. So hepcidin is part of the normal iron homeostasis, and it's produced by the liver, and it does two things. Iron is normally stored in macrophages, and then it needs to get out in order to go into the hemoglobin and go into the red blood cells. But hepcidin will actually prevent it and trap it from getting out of the macrophages. So we have enough iron, but it's not accessible. And then another thing that hepcidin does is it results in the GI tract absorbing less iron. So let's look at how we would identify it. Let's look at the smear first. A couple of key things. First is look at how similar these are all in size. So we have a normal red blood cell distribution width. They're relatively equal width. We have a decreased reticulocyte equivalent hemoglobin concentration. Now, we talked about that before as it indicates either anemia of inflammation or iron deficiency anemia. Why? Because in order to create a red blood cell, we need the ingredients, and both of these have limited iron. In this case, the limited iron is because the iron is not accessible. So because we're limiting the ingredients of red blood cell production, we have this decrease in reticulocyte equivalent hemoglobin concentration. So we're kind of stalling the process of creating the red blood cells because of the low iron, which is resulting in less hemoglobin concentration less relative hemoglobin concentration in the reticulocytes. Some other lab findings. We're going to have a low serum iron level. And we talked about before that the RET-HE is the fast test that we do. It might even already be included in the CBC. So that's fast. The iron concentration, that's something that we would do separately, and it would be a little bit slower. But if we have a decreased RET-HE, then we would do the iron panel. And we would see low serum iron for anemia of inflammation, but we would see a high ferritin level. Ferritin is a storage form of iron. And remember, there's plenty of iron, it's just not accessible. So different from iron deficiency anemia. Before we get into hemolytic anemias, I think it's important to first review what's inside of a red blood cell. We know that there's hemoglobin. There's also bilirubin. There's a molecule called lactase dehydrogenase, and there is G6PD. Now, if we lyse the cell membrane, we would expect that hemoglobin would be released out into the plasma, which is not a good thing. So we have haptoglobin that comes and captures hemoglobin. And so we would expect in hemolytic anemia to see a decrease in haptoglobin because it's trying to process all this free hemoglobin. We would expect to see in the blood work increased bilirubin, and specifically indirect bilirubin, because direct bilirubin would indicate a liver issue, but indirect bilirubin is an indication of hemolytic anemias. We would also expect to see an increase in LDH. And G6PD deficiency, we'll get into that a little bit later. That is referencing the lifespan of the cell and its ability to deal with oxidative stress. Some other key things that I want to point out here are that for mechanical destruction of red blood cells, we would see an increase in schistocytes. That makes sense, right? If we're running into things or we're running into vessel walls or whatever the case is, something's breaking these up, there's some friction, we're going to end up with these schistocytes. And for the immune-mediated hemolysis, we get increased spherocytes, which are these guys. And I'll show you one on here. This is a spherocyte. It's those little concentrated ones. And then for the immune-mediated hemolysis, we would get a positive DAT And this is also called a Coombs test. So again, we have a hemolytic anemia. What laboratory findings do we expect? Well, we know that we have LDH inside of red blood cells, and that's being released because it's hemolytic. So we will see an increased LDH. We will see an increase in indirect bilirubin, and we will see a decrease in haptoglobin because it's trying to process all of that free-floating hemoglobin, which can be very damaging. In addition to some of the other findings with anemia, in hemolytic anemia in particular, we have the splenomegaly, the enlarged spleen that can be palpable, and that's because there's so many fractured red blood cells that the spleen is trying to clean up the bloodstream and remove all of those little broken red blood cells and red blood cell fragments. The hepatomegaly, the enlarged or palpable liver, is a result of something that's called extramedullary hematopoiesis, and that means that you're having production of blood cells outside of the bone marrow. And it's sort of a backup mechanism, but it's trying to compensate for the insufficient production of red blood cells to meet the body's needs. So they're being produced, but we don't have enough to meet the body's needs. And then from this slide, I just want to point out the terms here. We have the macroangiopathic hemolytic anemia is due to contact with rough vascular surfaces, turbulent blood flow, and if you look at these are all like human-placed devices, so that's macro. And then for the micro, or the MAHA, you have shearing of red blood cells due to intravascular fibrin strands. So micro, think fibrin, and then macro, think all the things that we go in there to surgically place. And this is what that would look like on the right here. And you can see those schistocytes, they look pretty much like that cartoon we saw earlier, sort of like spiky and fragmented red blood cells. Something's breaking them apart. Hemolytic anemia can also be due to intrinsic red blood cell defects. These can be membrane defects, and that would be things to do with the ankyrin and spectrin protein or the protein complexes, the ability to really hold that biconcave shape. And if you have a different shape, then you have that reduced red blood cell deformability passing through capillaries. Even if it's like a spherical shape, you're going to have a less ability to tolerate going through the capillaries, and you might have that tearing and lysing. And then you can also have things like enzyme defects. G6PD is the red blood cell's ability to deal with the normal production of toxic radicals and mitigate them, and it can result in a shortened lifespan of the cell. And then you can also just have like a premature removal of the circulating red blood cells by the normal process that cleans up older red blood cells. It's just happening too early. And if we do have these things, we might expect to see increase in bone marrow production, increase of reticulocytes or other red blood cell precursors that might even be nucleated. When we look at the slides, as we can see here in this slide, we can see this red blood cell precursor here, and then here is another one of those little spherocytes. And so it's going to have, it's so jam-packed and it's so misshapen that it's going to have a tough time going through the capillaries without bursting. And a couple of key features here about hereditary spherocytosis, right? So it is hereditary, it's autosomal dominant, so you would expect to see others in the family with this type of anemia. And then spherocytosis refers to those smaller than normal, misshapen spherocytic cells. But if you look here, we have another red blood cell precursor. So we will find that the MCV, the mean corpuscular volume, is in the normal cytic range because it's averaging all of these put together. And then another key finding is the splenomegaly, which we would expect to see with a hemolytic anemia, right? Because the spleen is working overtime to try to remove all those fragments from the blood. The key finding here that we've now had multiple questions on is the elevated MCHC. If you have that elevation, you're going to suspect hereditary spherocytosis. But also, let's remember what happens when we burst our red blood cell. Remember, some of the ingredients in the red blood cell are the LDH and the bilirubin, the indirect bilirubin. So we would expect to see those elevated. She didn't actually include it in the slides, but it's in her lecture that the current test is actually the EMA binding test, and that is something to check for correct ankyrin and spectrin proteins. It will only bind to the correct versions, and in this case, it is an issue of these ankyrin and spectrin proteins, so they won't actually bind to hereditary spherocytosis. So if you suspect it, this would be the modern test, is EMA binding. Now moving into the G6PD deficiency. If we think about a normal red blood cell, go down here, a normal red blood cell is going to have a certain amount of G6PD, and I'm just going to make a little g here. And this is going to really determine the cell's lifespan, because a red blood cell normally just generates toxic radicals. So as you go through the lifespan of a red blood cell, you start using these up, and it can't make any more. So when you use all of these up, then that red blood cell can no longer survive. So everyone has to have some G6PD. You can't survive without at least some production of G6PD. But these G6PD deficient cells have a shorter lifespan because they have less of it. And normally, there's actually no symptoms. If we go back up here, most individuals are asymptomatic. There's specific triggers that are generating oxidative stress, and if we want to look at some of the common ones that we might be tested on, it can be triggered by medications, fava beans, certain foods, and infections. And then, oops, if we come back down here, if we have one of these triggers coming in, we're generating more toxic radicals, we're using them up all our G6PD, and eventually the cell will lyse. So there are certain populations that have, that we see more G6PD deficiency in, African descent, Mediterranean descent, and one of the reasons it's thought is because it's thought to be protective against malaria. It's also X-linked, so it's more common in males, and it is self-limiting. So as soon as we use up all of these G6PD, then we're going to have the lysing of this red blood cell, and it will spill its contents into the plasma. I want to go over what G6PD deficiency would look like in a slide real quick. And this is a picture from the slides. She showed us that these are blister cells. They just have these little blisters on the end. And down here, these are bite cells. Here it looks quite literally like somebody just took a chunk out of them. So these are showing oxidative stress, indication of maybe a G6PD deficiency. And then the Heinz bodies, right, if you see a bite cell, think of Heinz ketchup. This stain is specifically pointing out denatured hemoglobin. So if you have oxidative stress inside the red blood cell, you would expect to see denatured hemoglobin. Sickle cell disease is when you have a person that is homozygous for these sickle cell gene. If you have the sickle cell trait and you're heterozygous, you're generally asymptomatic. So sickle cell disease is homozygous for sickle cell. You get these elongated red blood cells, and they get clogged in the capillaries, and they can cause significant issues depending on which capillaries they get caught in. They can cause acute pain crises, like if they get caught in the lungs, for example. They can get caught in the hands. They can even get caught in the penis and cause priapism. They can cause a stroke if they happen in the brain. So just any small capillary, really, they can clog things up and back up the blood flow. And some of the things that we're looking for in the slide, in addition to these sickled cells, would be, if we look at the red blood cells, I'm not sure if you can tell, they have like a little dot. Those are called Hal-Jolly bodies. And then you can also see some target cells. We would want to do hemoglobin electrophoresis, and we'll look at the S column and the A column. And if you look here, we have plenty of S, but we are missing A. So we suspect this person has sickle cell disease because they appear to be homozygous for the sickle cell trait. Now we're moving into the microcytic anemia. If you see here, the mean corpuscular volume less than 80. This is often due to being iron deficient, resulting in defective heme synthesis. It can also be from like thalassemia and even anemia of inflammation. So I want to move down here and point out a couple of key features of iron deficiency. Is going to be this pica, which is the craving for non-food items, and also like the craving for ice. And then moving into what we would see in the labs and in the slide. So remember what we're comparing the red blood cells to, the nucleus of this lymphocyte. Look how much smaller they are. And in addition, you have some that are hypochromic. And this you would see a little bit later on in iron deficiency anemia, because it would take a little bit of time for the cells to become smaller, become hypochromic. But you have a low serum iron, and in order to compensate for low iron, the body is increasing transferrin. So we still have low iron, we have an increase in transferrin, and as a result, we have a decrease in the saturation of transferrin. So we have a high binding capacity for iron, but we simply don't have enough iron. And another key feature of the iron deficiency anemia is the low serum ferritin. Ferritin is an indirect measure of the storage of iron. So this is what we're looking for. We have low iron, we have low serum ferritin, iron storage. We have the body trying to compensate, so we have an increase of transferrin, but we still don't have enough iron, so we have a decrease in transferrin saturation. Our ability to bind iron, our total iron binding capacity, is high. We simply don't have enough. So hypoproliferative anemia. We're going to look at the retic count and see it's less than 2%. It's 1.5. So we have hypoproliferative anemia. And then some of the other key features, which she said that we're not really sure why, but we have a high platelet count. And we have a mean corpuscular volume here that is very low, very much less than 80. So we have a microcytic anemia. And very early on, you might have a normocytic anemia with iron deficiency anemia. But as you have that continuing malnutrition or whatever it is that's causing the low iron, then you're going to get into a microcytic anemia, and you're going to have the hypochromic cells. So laboratory studies, how do we distinguish between anemia of inflammation and iron deficiency anemia? Well, remember, in anemia of inflammation, we have iron, but it's being surrounded by macrophages and it's not available. So if we had, it's not really, we don't have the actual iron content, but in anemia of inflammation, we would have high iron. And in iron deficiency anemia, we would have low iron. In anemia of inflammation, we have a lot of iron, we have a lot of stored iron, so we have a lot of serum ferritin, but our ability to actually process the iron and use it is reduced. So we would expect this key finding in iron deficiency anemia, total iron binding capacity is low. There's not iron that's available for use. In iron deficiency anemia, well, we have a low overall iron content, which is causing a low reserve of serum ferritin level, but our ability to actually use iron, if we did have it, is high. So total iron binding capacity is high in iron deficiency anemia. Serum ferritin is low in iron deficiency anemia. And in anemia of inflammation, our ability to bind iron is reduced, but our serum ferritin level is high. The last of the microcytic anemias are going to be the thalassemias. They are a form of hemoglobinopathy. We already talked about sickle cell anemia. We have amino acid substitutions in the qualitative abnormalities that influence how they interact with each other. And in this case, in thalassemias, we actually have knockouts. So let's look down here. The degree of severity of thalassemia can range from silent to incompatible with life, depending on how many of the genes are affected. And we actually have 2 genes on each chromosome for a total of 4 genes. Some of the identifying features, and the reason that I'm showing you both alpha and beta together, are because we have some similar identifying features here, right? High red blood cell count for alpha and beta, normal red cell distribution width for both. We also have a lot of target cells. We also saw target cells before, didn't we, on iron deficiency anemia, but those were scattered target cells, and these are significantly more. So things that point us towards thalassemia: high red blood cell count, normal red cell distribution width, a high amount of target cells. So we suspect a thalassemia. Now what do? Well, we can do a definitive diagnosis. Is going to be molecular studies, and that's going to tell us if we have any gene knockouts. We can also do, in the case of beta thalassemia, we can do another hemoglobin electrophoresis. And if we see hemoglobin F, the fetal hemoglobin, if we have thalassemia major instead of just the trait, we're going to see a high amount of fetal hemoglobin in order to compensate for the missing beta genes. And another thing to highlight is it's actually diagnosed in the first year of life, as we switch from using hemoglobin F to HGA. So it's not going to be immediately apparent. So the last thing we're going to talk about is some of the additional tests, and a lot of these we actually already talked about. So if you suspect renal disease, then you would check your levels of creatine and erythropoietin. If you suspect anemia of inflammation, you're probably going to have a lot of C-reactive protein. That's an indicator of inflammation in the body. And we'll probably also have a high ferritin level, right? Because we have iron, it's just not available for use, which is kind of the opposite of nutritional deficiency. In the case of iron deficiency anemia, we're going to have a low serum iron content. We're going to have a low ferritin, which is the storage form, indirect measure of the storage form of iron. We're going to have a high total iron binding capacity, and we're going to have low transferrin saturation. If we measure low amounts of vitamin B12, that will also indicate nutritional deficiencies as well as copper. For thalassemia, we talked about hemoglobin electrophoresis, checking to see in the case of beta thalassemia if we see more fetal hemoglobin. And you can also do DNA sequencing if you're suspecting a knockout gene in one of the alpha globin chains. For sickle cell disease, specifically, we discussed the hemoglobin electrophoresis, right? If we have hemoglobin S instead of our normal hemoglobin A, then we would suspect sickle cell disease. And remember, sickle cell disease is actually the homozygous version, so we shouldn't expect to see any hemoglobin A on the electrophoresis. In that case, we'll just see the hemoglobin S. And then the hemolysis, remember, we talked about the general contents of a red blood cell, and these get released into the plasma when we have hemolysis. And because it's bad for hemoglobin to be released into the bloodstream, we also have decrease in haptoglobin, because haptoglobin is trying to mitigate hemoglobin in the just free floating in the plasma. So we're producing haptoglobin, but we're using it up, so we would expect to see a decrease in total haptoglobin. So that was my hopefully brief rundown of anemia. I hope it helps some of you guys as well.